Hi, last little Dr. Ben update for the day. This is about a paper. It's by Watanabe, and it was published in Science maybe a week or two ago. And this is looking at glycosylation. And so, what's that? All right. So, you've got your spike protein on the outside of the virus. That one we all know about. On the outside of the spike protein, there are actually modifications to some part of it. So, one of these common things that happens in proteins that are going to be on the surface of a virus or surface of a cell is that you add sugar chains to it, and this just happens all by itself in two parts of the cell. The ER is where they get added, the endoplasmic reticulum, and the Golgi apparatus, which is the coolest part of the cell to both say and draw, yeah, and color, yeah, because it looks like pancakes, yeah. The Golgi apparatus is where they get trimmed and modified and branched and made just weird and wonderful. And this is the first look at exactly what's going on with each of those. So, this paper figures out that there are 22 of these glycosylation sites because there's a particular amino acid. It's going to be an asparagine amino acid, which is where these things can get added. And on each of those 22, there are basically 22 different versions of a sugar chain. Some of them are huge, some of them are branchy, some of them are real small, some of them are like a primitive type or a more advanced type, and they're all slightly different. Yeah, isn't that wonderful? And just, like, what do you do with all this information? One thing that we know is that some antibodies, there's a famous one called, I want to say CR3022. But whatever, it's an antibody, you can buy it, and it's been worked out exactly where it binds. And it binds over one of the sites, and this sugar chain on the molecule is part of what the antibody is actually going to stick to. And so these things are both something that our body can potentially recognize and something that is also potentially going to block antibodies from being able to see certain parts of the protein. So it's like a camouflage for the virus, if you like. And yeah, it is a fascinating one. So among these 22, I count three sets of two that are kind of similar but not identical to each other. And so, yeah, basically, other than that, it's just a very, very wide range. This is information that we don't really have for a lot of proteins, and it's so cool that we know so much more about the coronavirus spike. This is going to be really important for how our immune system recognizes it. And if we can start to come up with ways to build in immunity that can attack this virus, understanding exactly how big and where and how many of these glycosylation sites are filled up is going to be pretty important in sort of deciding what it is we can throw at this virus that might actually stick to it and slow it down. So there we go. It's a pretty cool paper, and go look it up. And it's called Site-Specific Glycan Analysis of the SARS-CoV-2 Spike. Yeah, by Watanabe. Thanks very much. This is our Dr. Ben update for the day.